Hey friends, this is Fredrik again. In this video I'm going to talk about our work with TCRs, especially the ones where we're cloning human TCR sequences into expression construct for different purposes, being expressing them in cell lines in different ways, but also expressing them in mice. So let's see. So now I hope you don't see me. So here is a summary of the TCR locus. I think this could be just briefly summarizing, so you know how there is this is the TCR beta and this is TCR alpha, and it's going this stepwise process from the germline sequence down to the kind of ready mRNA. And you know that every time a T cell or a hematopoietic stem cell or a precursor cell decides to become a T cell, part of that process involves rearranging if they want to be a T cell, the T cell TCR loci. So there's the beta chain and the alpha chain. So starting up, if you look at focus on the alpha chain start, there's a lot of different V alpha segments, a variable alpha segments, 70 to 80 different ones. There's a lot of J segments, J alpha, about 61 here, and then there's constant domains here. And as the cell is developing, it will more or less random pick a variable domain and a V segment and a J segment, and get rid of a lot of the other ones so that they can kind of generate this other version, a rearranged loci or locus. And then it could make the mRNA, right? And here you know that if you look at the mRNA, you know that and compare the mRNA to the actual protein in the end, you know that there are certain regions that are making the interaction directly with the peptide MHC complex that's going to be on the cell up here, right? And there are certain regions here that are more important than others, and the most important one is the CDR3 region here, which is found both on the alpha and beta chain, which then is the border kind of between these regions here. And you see for the alpha chain, this is the border between the V chain and the J chain, caused the CDR3, and in the beta chain is even more complex because there's a V chain, a D chain, and a J chain. And by just combining different segments, then you get the very large variability in here. And I'm not going to go into the math there, but I think there is roughly 10 to the 10 different TCRs in the human adult body. The potential different TCRs that could be found are much higher. I don't know if it's 10 to the 18 or something like that. But randomly putting these things together generates this, the point being that generates a lot of variability here. And I should say this thing comes from this paper here. So what we are doing then is basing our studies on work by the Bettini lab and others, where basically have the TCR sequence, clone the TCR sequence from a patient, clone them into a retroviral expression construct, where this is going to actually be a construct that contains the human variable alpha, and it's going to be a chimeric version where we then have the mouse constant alpha chain. We're going to make a chimeric TCR alpha here, and then it has a P2A linker, and then it's the human viral beta chain, and then a mouse constant beta chain. And then there is a IRS and a freshen marker. So when this construct is introduced into a mouse, into a cell, it will be expressed. This will be expressed as one mRNA, and as you know, when it's translated, P2A will be cleaved. So you get both the alpha and the beta chain from the same mRNA, which means that you're going to get supposedly a 1-to-1 ratio expression of them, right? And then the cells taking up this will also get this fluorescent marker. And this could then be expressed in cell lines by infecting the cell lines with this construct, and then selecting cells that have the expression on the surface. But you could also infect hematopoietic stem cells from mice, and then graft those into mouse and generating what Maria and others are referring to as retrogenic mice, because this is a retroviral construct. I want to emphasize that there is the chimeric approach here, where having a mouse constant region allows for the interaction of the constant domain with the mouse CD3. So that is needed. So I'll come back more to this, but that is basically the setup. And as mentioned, this comes from this paper here. So let's now then go to a situation where we clone our TCRs, and I made a template here, but I'm hoping that one can use this template. One can just use other approaches to it. I think for when working more with it, it makes more sense to use a template. So just showing you what we're going to try to generate here. So basically here we have our this is an expression construct that we got from Maria. That then contains basically the variable domain from the patient. So that usually we get from the patient sequencing, we get this part here, which is going to be containing the CDR3. That's going to be in this region, so in the border between the variable and the V chain and the J chain. And then again, we have our mouse track, so the constant domain of the TCR receptor alpha, P2A linker, and then we're going to have a variable segment from the beta chain. Here's the CDR3, and there's also CDR1 and CDR2 in both of these that are also involved in interactions. And then we have our constant domain here. So this is basically what we're trying to do. And so the sequencing, I'm going to show you step by step, basically, where from the sequencing we get part usually from framework 3 of the TCR alpha into the constant region of the humans. We get this sequence, and similarly we get it then from the beta chain, something like this. Somewhere between 150 and 200 base pair, depending on the sequencing, is what we typically see. So what we're going to do then, if we get this, we're going to start by taking this sequence, and I'll show you this step by step. We're going to take this sequence then, we're going to identify which variable gene it is, and then we're going to, from a database, we're going to take this part here, and we're going to glue them together so that we get the sequence that looks like this. And that could sound a little bit, I mean, one can think about that taking a sequence from a database and trying to look at the functionality of a patient TCR. But the truth is that this part, the earlier parts of the TCR are typically not, they typically are germline, so they typically don't have a different sequence from the patient. But to be completely frank here, of course, we can't, since this sequence is not sequenced, we don't know. We can't say that this sequence that we put in there is the same the patient's going to have, right? But I never seen this part, so the framework 1, CDR1, 2, and framework 2, I never seen that being another sequence from a patient compared to the germline sequence of the specific V region. But I'm not saying it's never happened. It could happen definitely, and it might absolutely have relevance for the TCR. But since the sequencing material we have is this, we can't do anything else than adding this part here, and then we're also going to add a bit of sequence for cloning purpose, and then we're going to add the part from the constant, the master constant domain, actually down to here. So we generate a construct that looks basically like this, that has then cloning sites that we need, and then we do similar things for beta chain. So let's get to it then. So I'm going to use this template as mentioned, and then I'm going to use a couple of sequences, some examples. So this is typical type of sequence that we would get. So I'm going to copy that into our template, insert sequence here. This is going to be the TCR alpha. So I'm going to start by taking the sequence. So again, this is coming directly from the sequencer. And as you'll see, this will actually contain sequences that are related to the sequencing, the Illumina sequencing output. That's actually not from the TCR, but you'll see how I do it. So here I have a sequence. I'm going to copy that. Then I'm going to use IGBlast, which is a great tool. Make sure to have analyze T cell receptor here. I'm just going to paste it in here. Press search. And what you get then is identification of supposed different sequences here, or different regions. So this is TR-TRAV8/4 is the one that's has the highest e-values, the highest kind of significance. Actually, all of these three have the same, but all of them are TRAV8/4, and these are just a very high resolution of the sequence identity, which we don't really care about here, to be honest. So what you want to look at here is, and it also gives suggestion for which the J chain. So TRAV is, of course, T cell receptor alpha V 8/4. Then what you want to look at here is you want these E values to be fairly high. You want them to be, or low. You want them to be lower than -20, 25. Typically, they can go to 100 or something like that, depending on how long sequence. So you want to, but I always use the top one unless this C value is not good. You also want to just make sure that there is no stop cone identified, that things are in frame, and it identifies this as productive, right? And then you can actually see, so you can get things like this. It identifies the CDR3 sequence, and it gives you some other data. So this is a very useful tool. But what I'm going to use it for now is just that now we identified TRAV8/4. So the identified TRAV is 8/4. And then I'm going to go to ensemble. So this is ensemble human, basically. I'm going to search for the gene 8/4. So this is it, and I could spend a couple of minutes here just showing that typically the TRAVs looks like this: 2 exons, a 5' UTR, and the first exon, and then the second exon, right? And the exon just stops then. And this might look strange to you, but keep in mind that this is never going to be expressed only like this. It's always going to be spliced, spliced, but also to a J chain and a constant chain, right? C chain. So these are kind of fragmented a bit, which from if you use the regular genes and gene organization, they could look a little bit strange. But I just want to make one point here about the TRAV then. So this is human chromosome 14. And what we can see here, if you have 8/4 here, you can see that there's, as talked about earlier on, there's a lot of these different TRAVs, right? There's a lot of these different V segments that are just interspersed in this genomic region, which makes sense, right? I mean, we talked about that. There is 70 to 80 different of these, and what the cell does when it starts to want to become a T cell is actually to select one of these and basically get rid of most of the others. There's going to be some splicing also to get rid of some of them, but to a large extent there's going to be getting rid of these things here. So there you see. So here you see all the TRAVs, all the V regions of the T cell receptor alpha, and then actually here you see a lot of the J chains, right? So they just in proximity genomic wise. You also see that there is actually TCR delta chains here. So the delta and alpha genomic region is interspersed here. So you can find both there in the same region basically. But if you focus on the alpha chain, TCR alpha, of course, a T cell will only express either alpha or delta. So it doesn't. There's, as far as I know, never any problems with this. But they are closely linked, that's for sure. So the T cell will select an alpha chain. A variable domain of the alpha chain, a J chain, and then they will use the track, right? So the track here is the T cell receptor alpha constant, right? So they're all here organized like this, and you see there's a lot of J chains, there's a lot of and there's a lot of J chains, V chains. So this is how the locus looks like, which I think is kind of interesting. Anyway, so when we have the sequence here, 8/4, I'm going to export data, and then we get up the sequence here, and then we can see this is the cDNA. So here, but what we're interested in here is the CDS, and you also get data on the different exons and introns, etc. So what you get here, what I typically do here is to copy this, and the CDS, of course, is going to be the protein coding parts of the mRNA, of the cDNA, and it will start with ATG, so this is the start site, and typically it will end with a stop site. But here we're not going to have a stop site in frame, because, as mentioned, it's going to be spliced together or into this J segment. Anyway, you take this sequence here, I paste it into here. That didn't work well, so I'm gonna use something I use a lot, which is APE. So this is called, yeah, this is called APE, a plasmid editor, which is a great free software that is very simple to work with when it comes to copying and pasting. Because now I just paste it in here, and then it's gonna look good here. So then we have our sequence from the sequencer, and we have the genomic sequence, or the germline sequence. So if we look here, so we basically have now one sequence from the sequencer that's going to be something like this. But this is not going to be mouse tracked, and it's going to be human track from the sequencing. And then we have from the ensemble, then we're going to have this sequence here. So we're going to try to see where are they matching, and then we're going to put them together seamlessly. So what I'm going to do now is I'm going to take these sequences and I'm going to align them together to see where the overlap is. Align two sequences. Here comes the patient sequence. Here comes the ensemble sequence. And then we look for alignment, and we see there is a good alignment starting somewhere here. What you do see here now is that there is one mismatch here, and this actually happens. I said before that that we typically don't see that much difference, but there is actually, I mean, there's the occasional mutation found in these framework regions, which again, I mean, could be biologically relevant, but they could also be sequencing errors. Right? We typically go with this as much of the sequence as possible from the actual TCR that's been sequenced, right? But when we don't have any sequence, we need to go with the one that's in the database. So I'm gonna go for there. I want to redo this because I think there's an error here. ACCT. Yeah. Yeah, that was not the best type of example, maybe. I blasted again. Yeah, so here, so this is something that takes a little bit time to get used to it. But here, the fact that there is this doesn't align means that actually from here it starts to align. And so this is actually the sequence that where the kind of overlap between these two things are. I'll come back to these kind of things. So you're making some decisions when doing this, and now I've done this several times, so that's why I know that was an error, or how to choose that sequence. But what you'll see in the end is that I'm gonna put everything together and just make sure that it's correct in the end. Okay, so we see where there are overlaps. So then we want to introduce sequence A again. So this is what's coming from the sequencer. And then I'm going to put that into APE. And then I'm going to start to find this sequence and remove everything before this sequence. So find and then we find it there. And then I'm going to remove everything before that. And you see also that, yeah, I remove everything before there. And then I'm going to copy that and put it in here. And then I'm going to take sequence B. So this is the germline sequence, supposedly. Put it into ape again. And then remove the sequence identified in 5, this one, as well as all sequences after it. So we have like this. And then we're going to remove that one. And then when we paste these sequences together, they're going to be perfectly done in frame. Like that. And then if you look in the 3' end of the sequenced the sequence that we got from the patient sequencing, that is going to contain the human track toward the 3' end. And of course we want to make a chimeric version, so we need to kind of crop it there a little bit. So I'm going to paste that in here. And then I'm going to remove everything after this one. So this is the last thing I want to have from the patient. After here is going to be the human track, and this is actually what we want to make into a. Here is where the chimera starts, because actually this part here is also part of the human track. So the first part of the human track is actually going to be part of the construct, and then we're going to have a cloning site, and then we're going to have the mouse TCR. That's how the system is set up. So I'm going to put that in here. And then what we got here is actually a merge of all these sequences, basically. So what we have is we have 5' overhang for cloning. And then we're going to have so I go back to here. We're going to have the 5' overhang for cloning. And then we're going to have the part that is coming from the database, the germline sequence. Then we're going to have the sequence part, all of the variable domains, and so the V and J segment. And then we're going to have these mouse track, including a novel restriction site introduced there. So that is what we got here. So it's basically putting that together. So what we can take then to confirm this is we can I typically just copy this one into a new snap gene and just look. First of all, we want to make sure that we have our restriction sites needed. So we need that one for cloning, and we need that one for cloning. So they're both in there. We also sometimes use this EcoR1. So we know that the sites are there, and then we know directly after this site, so we should have the ATG. And if I identify this as a CDS, you'll see that the amino acid sequence starts, and there's no stop sites here. It just continues all the way, showing that the sequence is in frame. And just to be on the safe side, I also usually just look at, I know that the last amino acids here should be particular ones. And of course, if you want to search for amino acids, you need to change your find protein sequence. So I know that it's not only so that it's in frame, it's actually hasn't been any changes in that sense. The amino acid sequence is correct in it. So that's like the very brief things I do to make sure that this is correct. But I'll come back to do more things here. But so basically, we know now that we have this sequence 483 nucleotides, which is in the range that you would typically expect for these kind of constructs. And here we know that ordering these from IDT, that if they're below 500 base pair, they're cheaper than if they're above 500 base pair. So this is a good range. So then I'm going to go for my TCR beta here then. So TCR beta. I'm going to go here, TCR beta. Paste it in there. That thing we can remove. That was from something else. And then we put it once again into IGBlast TCR. I'm mentioning that because I think it's default that the IG sequence, so the immunoglobulin is the one. And when you can search for your TCR, and you will get some alignment. But first of all, you see that this is heavy chain, ID heavy V4, so it's not the TCR. And you see that you have these really low E values, sorry, very high E values. So remember I said you want to have this at minus 10 to the, or minus 25 or something like that. So this is, it's easy, since this is the default of choosing this one, this happens now and then you just get confused. So keep in mind to change to TCR. All right, so here we have TRAB-V, so T cell receptor beta V28 segment. Really good E-value here, really low. There's no stop colony, it's in frame and it's productive. Yes, it looks good, right? So I'm going to copy that one in here, like that. And then I'm going to extract the sequence like we did last time. Here we go. Again, they look something like this, 2 exons, most of them as far as I know, and they're localized close to other ones. I'm not going to go into more detail there this time, but they have a similar, the beta chain locus is similar to the alpha chain in many respects. Of course, we take the CDS of this one. I'm going to paste it into APE to get it to linearize it basically, and then we're gonna align them using BLAST2sec. To see where they overlap. So here we have perfect match. So I'm going to take it as part of the sequence. They start to align here. And then we're going to go back to the sequence coming from the patient. And we are going to crop insert sequence A below and remove everything before the sequence identifying 5. I'm searching for this one. Everything before here. And this might, I'm just, it could seem counterintuitive in a sense to remove a sequence from the sequence, but I know this sequence I know comes from the Illumina output, so this is part of the sequencing that is done there, and that could actually be good to validate in the sense that when you get outputs, you typically get many different sequences. You can align them and just see, okay, is there anything that's found on all of these that is likely coming from the sequencing, and here I know that that's the case. It's not depending on which software you use for or how you use to demultiplex your Illumina data or your NGS data from the sequencing. Of course, the sequence will look different if it contains this or not. But if it does, like it do here, you need to remove that, of course, because that's one part. And aligning them to several of the TCR output sequences that you get could be a good way of just seeing, okay, here is a repetitive sequence, so it's obviously not coming from the actual TCR. Okay, so then we done that, paste it in here. We'll go for the germline sequence supposedly, put it in, put it in here, and then I'm gonna remove the sequence, this sequence, as everything afterwards, like that. So I'm going to remove everything there. So now we know if we just put these together, they're going to align seamlessly. And then insert sequence C below and remove everything starting with AGG, ACC, TGAA. So I'm going to put that in here. And then I'm going to find here. So this is actually where the human track starts. So the J segment is going to be here, and then the human track starts, right? And we want to have a little bit of the human track, but then we want to put this chimeric type of structure as mentioned. And here actually it's just, here it says remove everything starting with. I did this a little bit, so here we actually want to remove this part, but actually this sequence is going to be introduced again here. So I paste it into here, and then again this is just gonna automatically concatenate different cells here. So it's gonna start with the 5' overhang, and then it's gonna go with the TCR alpha, start of the TCR alpha chain. Then it's going to go with the continuation of the, sorry, not TCR alpha, TCR beta chain in this case, the continuation of TCR beta chain coming from the sequencing from the patient, and then in the end it's going to have 3' overhang for cloning and start introduction this chimeric TCR. So we can do the same thing in 495 nucleotides again. This is under 500, which is not the end of the day if it's longer, but it makes a difference. So here we use MFA1 and BSTB1 for cloning purpose. So they're still intact, that's good. And here we know that the sequence is just gonna, there's no, since this is a P2A linked, so the ATG of this TCR beta chain, so now we are, basically we pasted in our construct from here to here that we just generated. The ATG, the start site, is actually going to be over here, right? So this is going to be expressed as one mRNA, and the P2A is going to be cleaved upon translation. So here there is no starting site. So we when we want to test if this is in frame, we just directly want to identify the first triplets as the CDS here. So we can look and see that there is no stop sites. It continues the whole way out, and then again, I oftentimes just make sure that there hasn't been any strange things happening. So actually that the last amino acids are the ones that are supposed to be fine. All right, so now what we do have a TCR alpha and a TCR beta chain, right? So what I want to do now is just to put this together as a construct, basically. So we have TCR alpha here, like that. Oops. Yeah, I need to paste it as numbers. So this is the TCR alpha. Then I should have prepared this, but then I need to basically take the part from the, so this is a construct that I made in the past. I should have prepared this. Just seeing how they start. But anyway, the point being that these are the sequences in the in between. So we start with our TCR alpha, and then we have, this is going to be the between sites, and then we're going to have our TCR beta sequence. That's going to be this one here. Yes, I need to paste it as text. TCR beta, which is going to end with this sequence. It's going to end with this sequence. And then when we cloned it into everything else, it's going to continue like this. So here we have basically TCR alpha. This is the sequence between the TCR alpha and the TCR beta, and then there's the end part of it. And then I'm going to use this concatenate to get them into one sequence, and then I'm going to make a copy that one. So basically, if we order these once and we make a construct out of it. Sorry, we order the TCR alpha segment, the TCR beta segment, and then we clone it into the expression construct, and the expression construct will look something like this. And again, just want to make sure that we have retained our SNAP-B1 restriction site. Often look at the EQR1, 2. We have our SAC2 sites, because those are needed to be unique. MFA1 we have the BTSB1 sites here. So those look good, and the software also identifies the P2A linkage, right? So here again, we know we have our ATG here, and when I define this as a CDS, this is what I think is the most important, right? So we really want this now to be a full amino acid sequence going all the way. So here is the TCR alpha part, then we have our P2A that, and then we're going to start have our TCR beta segment here. And it's going to go all the way, and our stop site is not until here in the end, basically, so, which is then important, right? So we as expected. So this is in frame being correct, basically, in that sense. So that is typically what I would do. There's more things you could do just to be on the safe side here. You could try to align this. So this is the final construct. We know that it's in line. I do oftentimes do a couple of different quality control steps. Make sure that it's an EQR. Yeah, I did that. We found that. We found that. Yes, so those are kind of things that you could imagine doing, like different quality control steps. We can take it and so we know that this is going to be, until here is going to be the TCR alpha chain. We can once again go to IG blast here. TCR, put them in here. And what we start seeing now, since we have, in contrast to in the beginning when I did this, I just used a truncated part of the rival domain, right? Here we use the whole one, so you start getting these really low numbers. But again, no stop codons in frame, productive, yes. So here we can now also start to kind of annotate things if we want. So this is the TCR alpha CDR3. We can have here, right? Yeah. We can say, if we want to, we could do kind of look at, for example, here we can say that framework 1 is going to be from 58 to 135, 58 to 135, 58, 135. Right, 5835. So if we want to, we can start to kind of annotate this thing. Here, of course, you could, just as mentioning here, you can see that there is a 27 base pair here, or 1, 2, 3, 4, 5, 6, 7, 8, 9 amino acids before the framework. So this is going to be the leader sequence. That is, so that's going to be cleaved off as far as I know. And I think this is something one could imagine actually changing between different TCRs. This is a concept I think I find it kind of fascinating that there's so many different variable domains, right, for the traps and for the TCR alpha and the TCR beta, the different V chains, and it seems like all of them have different leader sequences, and of course different leader sequences work slightly different. So I guess this is how evolution goes, but from an expression point of view, you could imagine. We typically don't do it because we try to keep it as normal as it you can argue that is not very normal, but keeping it as close to what we think is real, right? But you could, if you want, consider changing this leader to a really stronger leader sequence, for example. There are some that are more validated than others. Yeah. So this is the kind of things we could do. I do know that, so here's another one that we annotated using framework 1, CDR1, framework 2, CDR2, CDR3, looking at these different parts that you could do from this. But basically, so to me, this looks good in the sense that it is in frame and we find the kind of sequences that we want. You could do the same for the beta chain, right? Going in here, and to be really careful, I would also blast this versus the original sequence just to make sure that nothing has happened there. But in principle, this is the whole process as we do it, and then we would order the TCR beta chain sequence, TCR alpha chain sequence, and then when we get those, clone them into the expression construct. Yes. All right. I think that was everything I wanted to talk about. So thank you for taking the time. This is a long video. Bye bye.